Great. Thank you for the introduction. So my name is Rafael Levin. I'm one of Bruker Biosensors Application Scientists. And today I'm going to be giving a brief talk about the biophysical characterization of biologics and how we can take you from high-throughput screening all the way to cellular level analysis with our diverse instrumentation portfolio. If any of this is of interest to you, we are at booth 615, so please feel free to drop by. We'll be there until Thursday. So hopefully you're here because you're in the field of kinetics or you're interested in learning about kinetics. And what I'm showing on the left is two different interactions measured through SPR. One has fast kinetics, one has slow kinetics. But because the binding affinity is just a division of the off rate over the on rate, they would appear the same, right? If you do a traditional endpoint assay, such as an ELISA, you will get the same binding affinity, but you don't know anything about how fast or slow this is happening. In a complex world right now where there's different modalities for therapeutics, you might be interested in knowing, for example, how stable the interaction is between your therapeutic and your target. And so you might want to prioritize the off rate, for example. And that is precisely what we want to do at Bruker. We want to enable you with the tools necessary to be able to answer these complex questions. And so on the left, in the high throughput screening category, we have the SPR64 Triceratops, which is our highest throughput instrument. It is surface plasma resonance, label-free technique, very established for screening and multiplexing. And then more on the right, towards the lead optimization, we have our Helix portfolio, comprised of the Helix Plus, which is also great for binding kinetics, but has found a particular niche in the field of ternary complex formation through bispecifics or PROTACs. And on the right, we have the Helix Cyto, or our single-cell interaction cytometry, which is able to measure kinetics on the surface of live cells. And so the entire goal here is to give you more information early on before you even go into in vivo studies. So again, this is our highest throughput instrument, and I like to say we're strong in all 6 metrics on the left, but I want to just touch upon 3 of them. The first is the fluidics. So we have 8 individual needles that you can select or deselect for economic sample management or seamless assay optimization. We also have the rotatable manifold, which is a valveless microfluidics manifold, so it's not a consumable. It's designed to stay in the instrument, and it's part of the reason why we have the throughput. And then lastly, the plate handling and automation. If you work in a high throughput environment and you already have a robotic device, we are vendor agnostic, so we can easily couple that to our instrument. So just to expand a little bit more on the manifold, because it's a new development, you can see it here on the left. There is 8 inlets and 8 outlets, making 8 channels. And this is the part that physically presses onto the sensor to deliver your sample. So you can envision injecting 8 different targets in the vertical orientation, then rotating to the horizontal orientation. And at the intersection between the columns and the rows, you get your measurement spots. So we get 64 individual kinetic characterizations within a single experiment and within a single consumable. And you don't have to sacrifice any spots because between the columns, we have interspots, which you use as physical reference to reference out your data. So what types of assays can you do? All of the other assays that you can do in other SPR instruments, but I would just like to touch upon the multi-injection cycle kinetics, or MIC, where, for example, you can have 8 targets in the vertical orientation, rotated 90 degrees, and then bring 8 different analytes in the horizontal orientation with sequentially increasing concentrations of the analyte to give you an on rate and an off rate. So then this is how you get 64 individual characterizations within the single experiment. And this is actually what we did for an internal project. So we were trying to measure the interactions between Fc receptors and antibodies. So obviously antibodies are very complex biological molecules. And it's not only important to know what they're targeting, but also how they're interacting with other components of the immune system through the Fc portion. So with this assay setup, this is what we got from one of the experiments. We had 8 different Fc receptors in the vertical orientation, and then 7 antibodies with different Fc portions, as well as a negative control. And you can see that the negative control is not binding to any of the Fc receptors, while CD64 is actually binding to all of the antibodies in a similar way. So how can we relate this data to actual biological activity? So if you take nivolumab, for example, this is a well-characterized checkpoint inhibitor. So nivolumab binds PD-1 on the surface of T cells to prevent the interaction with PD-L1 overexpressed in cancer cells. And it's really good at doing what it does. But if you didn't touch the Fc portion, it might interact with other cellular components, and it could actually lead to T cell silencing. So what they did is they modified the Fc portion, and you can see that there's no binding to any Fc receptors except CD64, which was expected. You can also take CD16A, which has two naturally occurring polymorphisms in human population, a V form and an F form. And if you look at the rate scale plot on the right, while the associations are very similar between both forms against the antibody, the dissociation for the V form is actually much slower, giving it a tighter binding affinity. And this actually makes sense because the V form has more natural killer cell activity. So this is a perfect example of relationship between more binding affinity affecting biologic activity. So moving on to the Helix portfolio, I'm going to start by talking about the Helix Plus, which uses fluorescence as a detection mechanism. And for this, we have engineered a very novel biosensing chip where we have a gold surface right here and a DNA nanoliver covalently attached to it at the bottom. At the other side, one of the strands has a reporter dye. The other strand has your conjugated protein of interest. And so if you flow different concentrations of analyte, as you would do in SPR, you will see the dye having a quenching or an excitation effect. But for this talk, I would like to talk more about the Y structure. And the Y structure, as I said before, is a very elegant way to study ternary complex formation. So at the end, it's called a Y structure because it obviously looks like a Y. One of the strands has a green reporter dye, the other one has a red dye, and to each strand, you can conjugate a protein of interest. You can then flow a PROTAC or a bispecific, and you can either measure in both the green and red channels at the same time, or if the molecule is small enough, such like a PROTAC, you can go into FRET mode. So what we have here is an example of emicizumab. Emicizumab is a bispecific to treat hemophilia A, and it has to bring both factor 9A and factor 10 together at the surface of platelet cells to trigger coagulation. And what you see on the left here first is the bispecific against factor 9 alone, which gives us a sensogram, and you can clearly see from the dissociation that it's monophasic. We repeated the same thing with factor 10 in the red channel. And again, if you look at the sensogram, it is also monophasic. But then we conjugated both of them at the same time, and when you flow the bispecific, while for the red channel, the off rate and the on rate are the same, in fact, the sensograms look almost identical. For the green protein, now you have a slow off rate and a fast off rate. And you can clearly see that right here. So you have a fast population and a slow population, very different from what we got with the protein by itself. So this highlights that there's actually a ternary complex formation and there's avidity in play. Similarly for PROTACs, so we have the E3 ligase VHL, protein of interest BRD4, and the PROTAC AT1. And this was measured doing FRET mode. So if you look here at the left, we have the protein of interest by itself, monophasic dissociation from the sensogram. If you look at the right in the red channel, we have the E3 ligase by itself, monophasic dissociation on the sensogram. But then when you look at both of them in combination, the sensograms change a lot, especially in the green one. You can see, again, there's a fast population and a slow population. And if you look at the rate scale plot on the right, you can clearly see how much slower the off rate is. And again, highlights the ternary complex formation and avidity. And lastly, the question has always been there, is what you're seeing in the recombinant protein context actually replicating what happens in vivo? And that's how this technology was born. Single-cell interaction cytometry allows you to flow labeled analyte against your target of interest, but on the surface of native cells. So you preserve the native folding, the native density, the membrane mobility, and also co-interactors that you may or may not know about. And again, it measures in both the red and green channel, so it's a fluorescence-based technique. But now our biosensor has these flow-permeable, biocompatible polymer cages, which can physically trap cells. And it's cell type agnostic, so you can use suspension cells, adherent cells, primary cells, B cells, T cells. Pretty much, if it fits in the cage, we can capture it. And the workflow is pretty simple. First, you just inject your cells to trap it into the cage. Then you flow sequentially increasing concentrations of your labeled analyte. Then you inject buffer to get the dissociation. We're able to just do one dissociation because dissociation is not concentration dependent. And then lastly, you can reverse the flow to expel the cell. We've worked with all these analytes, with the most common projects being antibodies, nanobodies, and bispecific binders. But pretty much if you're able to label it homogeneously, we can work with it. And now I'm just going to finish up with some examples to highlight this technology. So here we have 3 different versions of antibodies against the same target. And just by looking at the sensorgrams, you can actually see that the kinetics are very different, while the antibody for control is not binding at all. And if you put them on a rate scale plot, you can see that while antibody number 2 has the fastest association, yield is the strongest binding affinity, it is actually antibody number 3 with the slowest off rate, the one that has the more stable interaction. So again, this goes back to the beginning of my talk where I said you might want to fine tune your biologic for a specific metric. We also know that there's really difficult to express proteins such as GPCRs with multiple membrane spanning domains. And what we have here on the left is a first step in the assay development where we were testing just a single concentration of a labeled small molecule against the CHO cell expressing the GPCR. So you can see some binding. And then there's no binding for the cell-free control or the cell not expressing the GPCR. And this allowed us to optimize the assay to do a 3 concentration titration and get a KD of 1.4 nanomolar. And then lastly, this is a manuscript that we have in preprint, just highlighting the technology a little bit more. So we have 3 different cell lines that all express low levels of HER2, but have different membrane chemistries. So some might have different glycosylation patterns, some might have different proteins on the membrane. And we had the same antibody targeting them. And you can see that the half-life of each of them is different. And in fact, it's much different from a cell expressing high levels of HER2 and also different from your traditional, I guess, more synthetic assays such as an SPR. So this just highlights stuff that you would not know otherwise. And that is it for me today. Hopefully, I was able to give you a brief introduction of how we can take you from high-throughput screening all the way down to cellular level analysis. Again, if you're interested, we're at booth 615, but I'm happy to take any questions at the moment. Thank you very much.